Hello, welcome you all to our family of BioGoogle. In the previous video, we have discussed about the structure of DNA. Structure of DNA that is proposed by J.D. Watson and Francis Crick. Okay. In today's video, we'll discuss about the functions of DNA. Okay. In different organisms, in different cells, at different phases of the life cycle of the organism, the DNA has various functions. Okay. Before that, we'll just briefly recall the structure of DNA, then we'll move on to the functions of the DNA. Okay. Firstly, the DNA is made up of two helically coiled structures or two polynucleotide chains, which are coiled in a clockwise manner or a right-handed manner. Coiled in a right-handed manner. Okay. At some place they leave the small gap. It's called a minor groove. At some other place they will give long gap. It's called major groove. And the total pitch of the one helix, this will make one helix. The total pitch of this one helix is 34 angstrom. 34 angstrom. And that includes 10 base pairs. That includes 10 base pairs. And the angle of rotation is 360 degree for 10 base pairs and 36 degree for one base pair. 360 degree for 10 base pair and one base pair, it is 36 degree of angle of rotation. And the diameter of this helix is 20 angstrom. It means it will coil only in the particular diameter. It will not go beyond the particular length, right? And one end of the one strand is known as a 5 prime end, other end will be 3 prime end. 5 prime because in the first nucleotide the sugar has a free phosphate group at the 5th carbon. It has a free phosphate group at the 5th carbon. So it is called 5 prime end. And the 3 prime end means the last nucleotide has a free OH group. The last nucleotide has a free OH group at 3rd carbon. Hence it is known as a 3 prime end. The other will be of course vice versa. Just because the two strands are anti-parallel in nature. The two strands are anti-parallel in nature. Anti-parallel means, of course, they must run parallelly, but the direction is opposite. If one is running in 5 to 3 prime direction, other will be 3 to 5 prime direction. Hence, they are referred to as anti-parallel strands. Right? I think it's enough for recalling the things. And in between that, the two strands are joined by the hydrogen bonds between the nitrogen bases. Adenine with the thymine, 2 hydrogen bonds. Guanine with cytosine, 3 hydrogen bonds. Like that, the DNA is been formed, and this model is proposed by Watson and Crick. Okay, very much important diagram in all of the examinations. Okay, either your PUC examination or a NEET examination or theory examination or else any other competitive examination, the diagram is very much important. And not only the diagram, the numericals with respect to the diagram, that is pitch of the helix, angle of rotation, and the number of base pairs. Like that, every numerical is important in this aspect. Okay, so coming to the main topic, today's video topic, that is functions of DNA. Okay, we'll see one by one in detail. Firstly, DNA performs, or it will carry, DNA carries genetic and hereditary information. DNA carries genetic and hereditary information. It means from parent generation to the offspring generation, maybe father, mother, son, daughter, etc. So the characters from parents will be transferred to offsprings only in the form of DNA, only through the DNA. We say chromosomes, but chromosomes ultimately contain DNA only. Chromosomes ultimately contain DNA only. So DNA is the genetic material that is responsible for transfer of the characteristics from parent to offsprings, or from one generation to next generation, next generation to next, like that. For subsequent generation, the DNA helps in transfer of the characteristics. Hence the first purpose is served very well by the DNA. Okay. Second one, variations. Here variation means parent offspring. Parent means father, mother. So genes from father, chromosome from father and mother will join together to form a offspring, baby, son or a daughter. Okay, but if it is son, he will not be exactly like a father. If it is a daughter, means if the offspring is daughter, the daughter will not be exactly like a mother. Daughter will not be exactly like a daughter. Means daughter is not exactly like a mother. It means there must be at least a few differences between mother and the daughter, or the father and the son, or parents and the offsprings. Of course, we can say some characters are exactly like a father or mother. You can say the nose is like father, the ears are like mother, the eyes are like father, something like. But there will not be 100% similarity between parents and the offsprings, just because during fertilization, after copulation, the chromosomes or the genes or the DNA from father as well as mother will combine 50% of the genes from father, 50% of the genes from the mother will combine to form a zygote, that will later on form the offspring. Hence, the offspring will not be exactly like any of the parents. The difference is known as a variation. And this is the basic material for evolution. Evolution means improvement of the characteristics or adaptation to the environment, changing environment. So as the climate changes day by day, maybe either due to pollution or any other factors, the organism has to adapt to that climate. So hence, in that situation, there must be the variations among the offsprings which are produced by the parents. That difference is known as a variation. Okay, that's only because of the DNA. The variation is only because of DNA. Right. Moving on to the next, mutations. Mutation are nothing but sudden inheritable changes in the genetic material. Okay, it is nothing but sudden inheritable changes in genetic material. Sudden changes, immediate change, either in the DNA, the number of base pairs may be increased, or any part may be deleted, any part may be removed, or any kind of mutation can occur. Mutations can be deletion, mutation can be addition, or mutation can be translocation, or mutation can be the inversion. We will discuss this in later classes in the same chapter. We will discuss all these 4 types of mutations in further classes. Okay, after 3-4 videos. Okay, we will discuss afterwards. So all these mutations are just because of the change in pattern of the replication or transcription, or any changes with respect to DNA. If the change is sudden, immediate change, that change is known as a mutation. That is also one of the basic for evolution. Mutation is also one of the basic material for evolution. Okay, so that's how the mutation is also because of the DNA or changes in the DNA or changes in the replication process or any mistakes in the replication process or any errors in the replication process. So all will lead to the mutation. Sometimes along with the internal factors, the change in environmental factors also lead to the mutations, like high temperature or any harmful chemicals. They will also lead to the mutations. Right. Next, replication. It means DNA can form another DNA. DNA can form another DNA. DNA by the process of replication forms another copy of DNA. Okay, the process is replication. The replication is also known as autocatalysis. Replication is also known as autocatalysis. Auto means self, same. Catalysis, any reaction, synthesis, preparation. So DNA is synthesized from DNA. DNA is prepared from DNA. So the same molecule is produced, same, auto, self. So it is replication is known as autocatalysis. Next we have a transcription. We have a transcription. In transcription what happens? DNA will form messenger RNA. So in replication DNA forms same molecule DNA, but in transcription DNA forms other molecule. DNA forms other molecule. So it is known as a heterocatalysis. Heterocatalysis. It is known as a heterocatalysis. It means from DNA RNA is formed. Means some other molecules are formed. It's not a DNA, other molecule is formed. Hence it is a heterocatalysis. So another function of the DNA. Next, recombination means during a meiosis, there will be a crossing between two chromosomes. One chromosome will be from the father, one will be from the mother. So in that process, the two chromosomes will exchange their genetic material. For example, I will just draw a diagram: chromosome with two arms, chromosome with two arms. So the two chromosomes imagine two chromosomes, one is paternal, one is maternal, father, mother, means chromosome from father, chromosome from mother. Okay, during meiosis, mainly the meiosis 1, prophase 1, during leptotin, zygotin, pakitin, in those processes, what happens is part of this chromosome will join the other chromosome, part of this will join the other chromosome. For example, after meiosis, these chromosomes will have, I will just draw a diagram to understand better. You can see here, this part of the maternal chromosome, this is paternal chromosome. So this part of the maternal chromosome has joined a paternal chromosome here, and this part of the paternal chromosome has joined here. So the two parts are exchanged. This process is called recombination. It's called recombination, also known as crossing over. Also known as crossing over. Okay, crossing over means exchange of the genetic materials or exchange of the part of the chromosome, or any small gene, a segment of the gene is transferred between or exchanged between the two chromosomes. This process is called crossing over or recombination. Because of this process only, there is a method called variation. There is a process called variation. Without crossing over, without recombination, there is no variation. There is no variation. There is no evolution. So for every evolution, recombination or crossing over must occur. Then only there is a variation. Then only there is a evolution of the organism. Right? That's about recombination. Then cellular metabolism. Cellular metabolism. What is metabolism? It means all the chemical reactions taking place in our body. Okay, some of all the chemical reactions. It may be catabolic reaction or it may be anabolic reaction. So all those chemical reactions in our body is known as the cellular metabolism. For that, there is a need of major biomolecules known as proteins. Major biomolecules known as proteins. But then how proteins are synthesized? DNA forms mRNA by transcription, and mRNA will form proteins by translation. mRNA will form protein by translation. These proteins are involved in metabolism. Proteins are a major part of the metabolism or all the chemical reactions, because these proteins can act as enzymes, proteins can act as hormones, or protein may also help in any other chemical reactions in our body. Hence, DNA is the sole molecule responsible for the metabolism, because DNA forms mRNA by transcription. And mRNA forms protein by translation. That protein will be involved in metabolic reactions. Hence, DNA is responsible for metabolic reactions. Okay, next, differentiation. Differentiation means when an organism, a cell divides into two, one cell will form two cells. Again, the two cells further divide to form 4 cells, 4 daughter cells. Again 4 becomes 8. So 1 cell, 2 cell, 4 cell, 8, 16, it will go on increasing. So from one cell, thousands of cells are formed. For example, a single cell, zygote will form an organism, or a baby. How is this? Is there only cell division? Just number of cells are increased, or more number of cells are formed by mitosis, meiosis? Then how the baby is formed inside the body from zygote? That's only because of differentiation. Means when some cells are formed, some 10,000 cells are formed, among that some of the cells will form one organ of the body, and few more cells will form another organ of the body. Organ 2, it is organ 1, and little more cells will form another organ of the body, Like that, a group of cells will form a tissue, then tissues will form organs. So the organization levels will increase. That's only because of the DNA. Okay. Next is the development. Means the overall development of the organism. Overall development may be the growth, reproduction, development, or any increase in size, increase in height of the organism. All depends upon the behavior of the genes. All depends upon the behavior of the genes or a behavior of the DNA. Hence, DNA is responsible for the development also. Then, very important, gene therapy. We will discuss in biotechnology. Gene therapy means there is a disease for any person. But nowadays there are techniques to identify the gene responsible for that disease. A particular gene which is responsible for a particular disease. So by gene therapy, we can remove the defective gene and add up a normal gene. We can remove the defective gene and add the normal gene. So removing the defective gene and adding the normal genes, the process is called gene therapy, or the method is called gene therapy. Okay, just replacing the defective gene by normal and functional gene. Normal and functional gene. Okay, so normal functional means the gene is taken from normal healthy person. One is this gene is present in the affected person, diseased person. But we have to extract a gene from a healthy normal person, and if we replace that gene, from healthy to normal, means healthy to abnormal or defective, the person or disease can be cured. A particular disease can be cured. Lastly, the major use of the DNA in forensic science is the DNA fingerprinting. Means if there is any dispute like paternal disputes, or there is any criminal investigation, for example, if there is any murder, so there is a sample of the blood in that dead person. So using that sample, they will draw some patterns of DNA. And of course, the police department will find some of the suspect. This is a sample available, and the department will find some of the suspected sample. Suspect 1, suspect 2. So by using the sample from those persons, again they will find the DNA patterns. So the suspect 1 may have the patterns like this. So the sample is not matching, means the DNA pattern is not matching with the suspect 1, so he is not the criminal. But when blood sample from this person is observed, the pattern was almost the same. The pattern, if it is found almost the same, it's matching somewhat, so he is the culprit. Like that, they will find paternal disputes, who is the father, who is the mother, to identify. If there is any orphan child without father, without mother, you can identify the father, mother by using the DNA fingerprint. We will study this concept at the end of this chapter. The last video of this chapter will be of a DNA fingerprinting only. Okay, so this is how DNA has thousands of functions for the living organisms. Maybe a plant, maybe an animal, maybe any microbe, any bacteria, DNA virus. For all those organisms, the DNA has got its own importance. Okay, I think this is enough for this video. Okay, if you get any more doubts, you can just add a comment in the comment section. Okay, I can come back to you with the answer for you, right? And if you are visiting our channel for the first time, just please press on that bell icon, press on the subscribe button. So please subscribe, just look at the red button, press that red colored subscribe button, and when you press the subscribe button, you will find a bell icon. bell icon. So please press on the bell icon also, so that you will get notifications whenever I post new videos. Okay, better, there's no loss if you just press on that. Please press on that. Okay, people just watch out the videos and move out without subscribing to the channel. Okay, you are not paying for that, just you are watching the videos, just taking the benefit of that. So please subscribe and press on the bell icon, so that you will get notification. And if you like the video, please share the video to your friends. Okay, whoever, if you are a teacher, share to your students. If you are a student, share to your classmates. Okay, so we'll see you in the next video. Till that, keep learning and keep watching our videos. Thank you, and Jai Hind, bye bye.